I'm working on embryos, and the big fascination about embryos is that they develop in an egg, and the egg is very simple. It looks very simple, and there are no structures in the egg which resemble in any way the animal which will develop in it. And this is a fascinating property. And in embryonic development, when you can observe it, you can follow it in certain animals, how it gets. The cells start dividing, and they get more and more and more, and then all of a sudden they start taking on shape, that they organize each other, they sort out, and they fold into the interior, and then they start forming an organism. At the beginning it's still very simple, there's only a few cell types, and they are arranged in a reproducible manner that is in each individual embryo in the same animal. They look very similar, and then they get more complicated, and they form the different structures which then later make the animal. And in normal development, the egg contains all the nutrients such that a shed egg is able to form an animal, and the animal will crawl out of it. It's either a caterpillar, or it's a maggot in the case of flies, or it's a little tadpole in the case of frogs. Or also in fish, in the case of fish, there's some larvae, you call that larval stage. So what comes out of the egg is not yet the adult form, it's not yet the grown-up animal, but it's a small form which you call juvenile. And this is already able to feed on its own, and then somehow to thrive, to grow, and then it gets an animal. And then the animal can multiply again by making eggs, and these eggs are fertilized by sperm, and then the whole process starts over again. So what we are interested in is how the eggs are organized. So I mentioned that they are full with nutrients, with yolk, and they also have some sap in it, which contains lots of factors which are swimming in it, and then they all have the nucleus, and the nucleus contains our genes. And the cells divide. So the egg cell divides into 2 cells, and then into 4 cells, and then into 8 cells, and then into many, many, many more cells. And in each division, the nucleus divides first, and with it all the genes. So each daughter cell gets the same genes than the mother cell. And it means that the embryo is in fact a clone of genetically identical cells. So all cells have the same set of genes. They develop from one fertilized egg cell, and they genetically are all the same, but then later they all look different, and the secret is really to understand why they get different, although they have the same genes. And the genes make all the proteins in the cell, and somehow the cells must differ with respect to which proteins are made in which cells, and this means that it depends on which genes are expressed in these cells, that is, turned on or off, which genes are required to make the products. And this is really something which is quite fascinating, and it starts early by some factors being, although you can't see that, people can make it visible by sort of painting different molecules, so you can see, or you can show that there are molecules which are distributed in a non-random fashion in the eggs, and say they are polarized. Some factors are concentrated in one side of the egg and not on the other, and then the cells into which these factors, which are concentrated at one side, come to lie in, they will then develop in a peculiar direction and not in another direction. And in this way, in subsequent steps, other cells also get maybe influenced by neighboring cells or so on. They also receive different factors from other cells, and this is how cells gradually get different, and in principle finally end up in making this complexity of an organism with lots of organs at particular places, and the appendages at the right place, the head at the front, and the tail at the rear, and so on. And this is a quite fascinating problem, and perhaps it's the most complicated or complex problem in biology, because it means that from something simple you get something complex, which is in contrast to what we learn from the rules of physics and chemistry, that you always get from the complex to the simple. This is the easier way. This is the normal way. And why is it different in life? Life has this property that complexity arises from simplicity. And this is just a fascinating thing. And I mean, it's really not so easy to explain that in 5 minutes, but we probably cannot say that we understood it, but in principle it's probably that in the genes we have all the information, and the information has to be realized, and it's sort of stored in the nucleus, and it's realized by some genes being then making their products, and the products then influence other genes, and these genes then influence other genes, and then you get a gradual separation in time and space of these different products, and so cells get different, and they keep on getting different, despite the fact that they want to get simpler, in fact. And so if they would die, they would immediately decay, and nothing would happen anymore. Decay also occurs in normal development, and an important factor in development is not just cells dividing, growing, and taking on different shapes, but also sometimes cells are born and made, and then they die later. And there is another process, in an interesting process in development, that cells are not only be made to do something different, but also sometimes many cells are made, and some at particular places will die. And so, for example, the fingers are made that way. It's first a big pad of cells, and then between the fingers, cells die, and then you get these protrusions. This is one way of making fingers. And another way of making, I mean, fingers are different from how you make an arm. An arm is made by a little bud, so it's a little bubble, and then from this point on, cells start growing, and they make a protrusion, and then it gets a long extension. And this is the way an arm is made. But the fingers are made by just killing of cells dying between the individual fingers. Yeah, and other processes which we are also now studying is a little bit later in development, when cells start migrating from particular places of origin. They're born at particular places in the body, and then they migrate long distances in order to make connections between different parts of the body. And this is, for example, the way nerves find their way. Nerves are extensions of nerve cells, and they make connections to distant parts in the body, and then they are the ones which can guide. They're pathways where irritations or sensations can be passed back to the brain, and they can make the connection between sensory organs and the brain. And this is also a very important process. We are studying it in the fish embryos, because the fish embryos are transparent. We can watch them in live form under the microscope. They develop under our eyes, and we can see, we can follow these extensions. We can follow the nerves as they grow out. We can label them with particular dyes which wouldn't harm the embryo, and which we could see, and then we can trace the cells in the living organism.
So actually all the future of interactions in the future of the nervous system are already seen in the embryo development.
Not all of them, but it's also developing gradually. And there are very simple connections first, and then there are more complicated connections later. And some connections are made by nerves growing out of the brain to find particular places in the body, and they make connections between these places of the body to the brain, like the eye and the brain and the nose and the brain and so on. And sometimes it's really groups of cells which migrate to the body and pull the nerves with them, and then you also make a connection.
And the shortest connection is between the eye and the brain, or nose and the brain, or?
Eye and brain is a pretty short connection. And this is very interesting, the eye and the brain. I don't work on this, but this is a very interesting process, because the eye has a It's like a camera.
Yeah.
At the end, at the back, it has this retina, which is like a film on a camera, and then you have the connection of the points which you see to this retina, and it is a mirror. It's a picture at the back of your eye, and this picture has to be transformed somehow into the brain in the same order. But the brain also has this figure, and this is a very interesting process how you get this connection. And there are these nerves from particular parts of the retina, which is the back of the eye, to reach particular points of the tectum. And you have another image in the brain. This is called the tectum image.
Mm-hmm.
It's the part of the brain where the image from the eye is ending up. And this connection must be very precise, otherwise your picture will not be seen properly. And it's not easy to understand how this is made. And you can do experiments there, and people have done experiments, and they found molecules which are present and guide these nerves to come the right route. But it's very complicated.